Good afternoon, everyone, and welcome to a webinar to discuss the 3-year data from the EMBARK study of ELEVIDYS gene therapy. We're really thrilled and pleased that you've taken time to join us this afternoon on this snowy afternoon in Ohio and the East Coast. Hope that you're warmer. We're really thankful to learn about the 3-year data from the ELEVIDYS gene therapy study. And with us today, we have James Richardson from Sarepta, who's the Chief Medical Officer there, Wendy Erler, who leads advocacy for Sarepta, and we have Dr. Crystal Proud from King's Daughters in Virginia. So with that, and without any hesitation, I would like James and Wendy to take over and start going through the slides. We will follow this with a question and answer period at the end. Should you have questions, please put them in the chat. We already have some questions that were recruited by the community as well. So together we will go through all of our questions, and following that, in a few days, we'll post this webinar as well. So welcome, James, welcome, Wendy, and welcome, Crystal, and let us begin.
Thank you so much, Pat, and thank you to PPMD for organizing this webinar. It was really important to Sarepta. We had an investor call earlier today, and we wanted to get this information to the community as quickly as possible, recognizing how important all of you are to the development program for ELEVIDYS, and to make sure you had an opportunity to hear this information from us as well. Thank you, Dr. Proud, for joining. So of course, I can't advance the slides here. As Pat said, I'm going to be joined by James. I'll do the opening and then turn it over to James to walk through the clinical and scientific data from the EMBARK clinical trial of ELEVIDYS, and this is 3-year top-line results. This slide is really important. The purpose of this webinar is to provide general education and scientific information in an update forum. It is top-line information, literally hot off the press. For all personal healthcare decisions, families should work with their clinical care teams. The information we will share today reflects what we know as of this time and may change over time as additional data continues to become available. We're going to talk a little bit about the really important safety information and some updates to what is called the ELEVIDYS label, which is the prescribing information that sites and physicians go by when they're prescribing ELEVIDYS to patients. This important safety information comes, as you may know, in November of last year. FDA updated the ELEVIDYS prescribing information, including adding a black box warning, which importantly calls out the risk of acute liver injury and acute liver failure and additional safety monitoring. Also, the medicine is now indicated for non-ambulatory patients 4 years and older, and the new label highlights enhanced safety measures. In addition to the prescribing information, FDA issued an ELEVIDYS medication guide. This is specifically designed for families and for families to have an opportunity to talk to their care team about considering ELEVIDYS. The purpose of reviewing this important safety information, or ISI, with families is to ensure that you all understand critical risks, side effects, safety measures, and anything associated with the product, ensuring that you have the most information available and can make safe decisions and prevent harm. Specifically to ELEVIDYS, the decision to receive ELEVIDYS are made with the treating physician, who will talk with families about additional precautions and what to know, including risks of infection and being aware of cardiac monitoring. On the infusion day, patients are monitored closely, and any concerns should be reported to the treating physician immediately. Treatment for patients with high level of antibodies to the vector is not recommended. And finally, it is important for families to discuss both potential benefits and risks to treatment with their physician when making any treatment decision. I mentioned a few minutes ago that part of the resources that are available is the FDA medication guide. That's available on the website shown here. Also, more resources can be found at elevidys.com. ELEVIDYS is only administered at specialized medical centers and hospital settings that are specifically trained and equipped to handle gene therapy and manage everything required for the infusion and care after dosing. More information, as I said, and additional resources are available on elevidys.com. Now I will invite Dr. James Richardson to discuss the 3-year top-line EMBARK phase 3 clinical trial updates with you.
Thank you, Wendy.
And as a minor disclaimer to begin with, it's a snow day here in Massachusetts. So if I am rained at any point by my children, I must apologize. But nonetheless, it's a great pleasure to be here, snow or not, to share the newest results from the patients dosed at the start of the EMBARK phase 3 study. So overall, these new 3-year results show that patients who were treated with ELEVIDYS in EMBARK Part 1, who are aged on average 9 years at this assessment, demonstrate a substantial shift in their disease trajectory and a treatment effect that has increased over time. On average, if we use the combined measure of physical function that I'm sure many of you are familiar with, the North Star Ambulatory Assessment, or NSAA, the ELEVIDYS treated patients actually remain above their starting value or baseline 3 years after treatment. In addition, ELEVIDYS patients have a 70% or greater reduction in their expected decline in the time they take to rise from lying on the floor, that's the time to rise, or the time to run or walk 10 meters. These are the 2 timed function tests that we assessed in these new data. These time function tests are highly predictive of future decline in the disease. This long-term information was collected from a well-run and carefully controlled phase 3 clinical study, and it shows the impact of a therapy that is designed to address the underlying cause of Duchenne. I think it's fair to say that what we're seeing is not only meaningful, but that it gets stronger over time, creating a sustained and growing separation from the expected trajectory, the known inevitable decline that we see in Duchenne. Next slide, please. Understanding the untreated disease trajectory and the potential outcomes based on when you treat are important. In Duchenne, as you know, the course of the disease follows a pretty well understood and unfortunately predictable path of decline, as shown here with the yellow curve that follows the North Star Ambulatory Assessment. Individuals with Duchenne do gain skills as they grow, typically reaching their peak physical function at around the age of 6, but then decline as a result of increasing scarring and loss of muscle. You can think of this as sort of 2 forces acting against each other: normal physiological growth pushing up, and then the impact of the disease pushing down. The aim of a dystrophin restoration treatment like ELEVIDYS is to try to relieve this constant downward pressure on the development and maintain and help the younger children acquire more skills. This, again, I think really highlights the importance of early treatment with dystrophin restoration, just to try to relieve this pressure and slow the progression. Depending on where on this curve an individual with Duchenne is treated, the benefits you see may look a bit different. So the benefits here are modeled with these blue lines. And so younger individuals, such as the line on the left here, may reach a higher peak than expected, for instance, whilst older individuals, those blue lines more to the right of this curve, may decline less slowly than expected. In all instances, it's really important to find an appropriate comparison matched to the treated individuals so we can best understand the effect the drug may be having. Next slide. So we have the most comprehensive data set in Duchenne drug development and commercialization with greater than 1,200 patients treated with ELEVIDYS, either in clinical trials or in commercial use. Next slide. We have or are conducting multiple clinical trials across a very wide range of individuals from the age of 2 up to the age of 24, and from weights as low as 12 kilograms up to 90 kilograms. Next slide, please. So we're going to talk today about the 3-year results in patients dosed at the beginning of the EMBARK study. So as a reminder, EMBARK was a double-blind placebo-controlled trial which had a primary readout at 1 year following treatment. At the end of this first year, the placebo patients crossed over and received ELEVIDYS, and the patients who had already received ELEVIDYS got a placebo infusion. All patients at this second point got to the per protocol increase of corticosteroids that participants get, and you get in the commercial setting around that time of infusion. Just to understand that without a placebo arm, so all those placebo patients are treated at the end of the first year, any subsequent comparison that we make to treated patients will require a new external control. We no longer have a placebo control to compare to. At the end of the 2 years of the EMBARK study, individuals were offered the option to continue in a long-term clinical follow-up, a study called Expedition or Study 305. And this will follow patients out to at least 5 years following infusion. So today what we're sharing is a 3-year data from those patients dosed at the very beginning of EMBARK in part 1 and are now enrolled in this long-term extension study 305. Next slide, please. So before we look at the 3-year data, here's a reminder of the functional results for all the patients treated in part 1 after 2 years of treatment. As you can see here, so this is a forest plot. So all the different tests are in the lines. Those blue boxes are different tests: the NSAA, the time to rise velocity, the 10-meter walk run time, and the 10-meter walk run velocity. And then that red line marks essentially equipoise between treating and not treating. And so everything that is to the right of that line means that on that outcome, on average, patients did better on treatment than compared to the external control. And those bars to the side of the black or blue squares, sorry, back a slide, Wendy, they're the margin of error for each of these measures. And so what we can say, because all of those dots consistently, all those squares lie to the right of that red line, on all of those measures at 2 years, there was benefit for ELEVIDYS treatment versus the external control. And all of those measures reached statistical significance, meaning it's very unlikely that any of these results were due to chance. So these results were just published in the peer-reviewed medical journal Neurology and Therapy earlier this month. Next slide, please. We've also previously presented data on muscle MRI. So muscle MRI, so that's images of the muscle taken within an MRI scanner, provides a direct approach to assessing muscle health. It measures, amongst other things, the amount of fat that infiltrates muscles. So as the inflammation of Duchenne progresses and muscle cells are broken down, they're replaced by fatty cells. And the level of that fat can correspond to the health of the muscle and can also correspond to how well that individual is able to use that muscle and perform tasks like climbing stairs or getting up from the ground. Lower levels of fat fraction, or that fat infiltration, indicate a healthier muscle. So this is the data from 2 years of EMBARK on muscle MRI. We don't have an external control yet for MRI, so I'm showing the 2-year data, that's the purple lines, and then the 1-year placebo data, that's the gold line. We see no increase in fat fraction from baseline to 2 years, and at levels at 2 years well below what is seen at 1 year in the placebo patients. This is consistent across muscle groups. Even though you see that improvement in NSAA and other functional measures on the previous slide, you see here that the impact of treatment being delayed even 1 year is damage in the muscle and an irreversible damage, and you'll never achieve the same level of fat fraction as patients treated a year earlier. We're very pleased with the treatment effect we see in both the blinded study functional outcomes and that reconfirmation with MRI you're seeing here. And I think this really highlights the importance of treating as soon as possible to preserve muscle. Next slide, please. Over the past year, we've also presented and published data on the heart taken from cardiac MRI scans over 2 years in EMBARK, which have shown no new signals and heart function remains in the normal range. Next slide. And the next slide. So I'll now get into the new 3-year data from EMBARK Part 1, treated individuals in greater detail. So as a reminder, these pre-specified or pre-planned analyses take data from the complete 2 years of EMBARK and the first year of our long-term extension study, Expedition. This is by far the largest long-term follow-up of patients with a gene therapy for Duchenne, all conducted within the rigor of a clinical trial setting. To understand how well the treatment worked for the boys in our study, we compared their 3-year results to a large group of children with Duchenne who had not received ELEVIDYS. This comparison group is created using a scientific method called propensity weighting. In simple terms, it helps us create the fairest possible comparison by matching each treated child with children from the real world data who are similar in age, abilities, and other important characteristics. We focus on 3 key measures of physical function. The NSAA, as said, a combined score of many motor skills. Time to rise, how quickly a child can rise up from the floor. 10-meter walk run, how fast a child can walk or run 10 meters. These measures are widely used in Duchenne research because they reliably show how the disease changes over time. They were also the main outcomes we plan to evaluate from the beginning of the EMBARK study. The external comparison data comes from carefully selected natural history studies and placebo groups from recent past trials. All of these children were receiving the current standard of care, including ongoing corticosteroid treatment. So choosing from a pool of more than 1,200 children not treated by ELEVIDYS, we selected those whose baseline characteristics closely matched the participants in EMBARK. After selecting that group, we applied some additional statistical adjustments to make sure the comparison was as accurate and fair as possible. This process really helps us understand with greater confidence what differences are truly due to treatment. Next slide, please. So this approach allowed us to create a very close and fair comparison for the boys who received ELEVIDYS. On the left side of the slide, you can see how well the 2 groups match. And this is how they look at baseline. So this is before they were treated with ELEVIDYS, or at the beginning of the external control data that we choose, so 3 years prior to the end of that analysis. On the other side, you'll notice that we had a large number of children in the external control group. It can often be a limitation of these analyses that you have small numbers to compare to, which makes your conclusions less certain. Here we have really, I think, quite large numbers, which help make the results stronger and more reliable. You may also see that 11 children from the EMBARK study did not continue into the long-term follow-up study Expedition. We looked carefully at these cases, and there's no indication that their decision to stop participation affected the results or introduced any bias into the results that we're going to see. Next slide. Keep us waiting, Wendy. So when we look at the NSAA scores, which measure these day-to-day motor skills, we see that the boys who receive ELEVIDYS are doing meaningfully better 3 years after treatment. On average, their scores are more than 4 points higher than we see in the comparison group of boys who did not receive ELEVIDYS. And this difference has doubled between years 2 and 3. This difference is not only clinically meaningful, it's also highly reliable from a statistical point of view, as you can see by the very low p-value. Even if we look at only the boys who received treatment, we see that as a group, they're still performing above where they started 3 years earlier. They are above their baseline. Now that's really important because in Duchenne, this is an age. So again, these patients are on average 9 years of age at this point, where motor skills usually begin to drop quickly. At this stage of the disease, most children typically show a clear and often rapid decline in the NSAA. And we can see this clearly in the external group, who lost almost 4 points over the same 3-year period. Next slide, please. Now let's talk about time to rise, which measures how quickly a child can stand up from the floor. This test is one of the earliest and most sensitive ways to see changes in muscle strength in Duchenne, and it's closely linked to predicting when a child may have difficulty walking. The sadly relentless decline of Duchenne is demonstrated by the progression of the untreated individuals, whose time to rise, which at baseline was only 3.5 seconds on average, has increased by over 8 seconds in the 3 years since the analysis began. The resultant difference of 6 seconds between the control and the treated patients is again highly statistically significant, with a p-value of less than 0.0001. If you're not very familiar with p-values, this is a very low one, indicating these results are very, very unlikely to be due to chance. This difference between treated and external control represents a 70% reduction in the expected decline in this ability, with the treatment benefit growing 3 times between years 2 and years 3. So you see that change of 2 seconds at year 2 is now 6 seconds at year 3. This reinforces what we saw with the NSAA, that by stabilizing the disease, the size of the treatment benefit can grow over time. Next slide, please. So this is the results for the 10-meter walk run. As with the NSAA and the time to rise, we observed a highly clinically and statistically significant difference of 2.7 seconds, again with a very small and statistically significant p-value. Again, this represents an approximately 70% slowing of the expected decline in this ability. And again, we see the widening treatment effect over time, with a doubling in the separation between the treated population and untreated population between years 2 and 3. So here at year 2, you see the differences 1.36 seconds, and then at year 3, the difference is now 2.7 seconds. Next slide. We also looked very closely at safety over the full 3 years. We have presented and published these data out to 2 years. What is new today is the column on the right, the year 3 data. So importantly, we did not see any new safety concerns during this time, and there were no serious side effects related to treatment in year 3 for these Part 1 treated individuals. This is consistent with everything we've learned about ELEVIDYS from more than 1,200 people who have received the drug in either clinical trials or in commercial use. Next slide, please. So in conclusion, this 3-year data from EMBARK and the Expedition study represents by far the largest long-term follow-up within the well-controlled clinical trial setting that we've seen for any gene therapy for DMD. Using a pre-planned analysis, the results show, I think, definitive and growing benefit to treatment in patients who are now, on average, approximately 9 years of age, a time at which the untreated natural history is one of significant loss of function. On NSAA, in stark contrast to the predicted trajectory, patients remain above their baseline 3 years following treatment. Individuals also showed a 70% reduction in the expected decline in the time they take to rise from the floor and the time to walk or run 10 meters. Equally important, there were no new safety signals. Long-term follow-up will continue, and we'll continue to share our findings with the community. While the data provide us, I think, with powerful information to inform changes in how the disease is treated, none of these results are possible without the individuals and their families who participated in our studies. We are incredibly grateful for all of your contributions, along with the contributions of the investigators and site teams and my fellow Sarepta colleagues who continue to inform our understanding of Duchenne and ways to change the course of this terrible disease. That finishes my presentation. I'll hand back to Pat.
Yeah, thank you, James. Thank you very much for that presentation, and Wendy for your introduction. It's very helpful, and these are really interesting and welcome results to our community, as you might know. I think it does reinforce our understanding that 12 months is a very short time to see change and to be able to have data that identifies change in this disease that is progressive. So I'm going to I was going to go backwards in the slides just because it might be easier with my notes, but a couple questions, and now welcome also Crystal Proud to talk to us, and James and Crystal, I think you'll do this tandem back and forth about answering questions. So one of the questions that has come through is how early and how are you thinking about treating infants? I know it's not part of the study and was not part of this study, but as we go back forward, the questions, are you thinking younger? You've mentioned at the outset that you've treated patients from 2 years old to 24 years old. So are you thinking of dropping lower in future just as an opening?
So obviously we currently labeled 4 and above in the United States. We have, as you highlighted there, Pat, conducted clinical trials down to the age of 2. We've seen some really, I think, expression data in those younger individuals and I think good safety. So we are, I think, very interested in doing further conversations with the FDA in the hope that we can offer treatment earlier.
And then if you don't mind, I'm going to start at slide 25 and go backward. In your conclusions, James, and certainly in the intro from Wendy, we talked about the black box warning on the label. And one of the questions that has come up repeatedly is the use of sirolimus or sirolimus, however we would like to pronounce it today. And are you now going forward in the commercial arena using sirolimus frequently, or with recommending it in the commercial setting, or is this up to the clinician? And maybe James and Crystal will comment.
Certainly, I can start, James, if that's okay. I think that there is ongoing use in the clinical setting of sirolimus in hopes that we will see a reduction in adverse effects on the liver. And we had some nice data that was presented by Dr. Soslow at the World Muscle Society Congress last year that demonstrated that it appeared to have a lower incidence of liver inflammatory changes when sirolimus was used as a medication just prior to the gene transfer therapy. We're taking that a step further, and we will be starting this next cohort within a different study, looking at older and non-ambulatory patients in particular who will receive sirolimus a couple of weeks before they receive gene transfer therapy with ELEVIDYS, and then we'll see how they do over time. And hopefully this will demonstrate that this is an effective way for us to reduce the potential for liver toxicity.
Crystal, can you just clarify what sirolimus is?
Yes. So thank you. It is a medication that is taken orally. It comes in both a liquid formulation as well as a pill formulation. And we utilize it to modulate what's called a T cell response in the body. And so it helps to reduce the immune system's response to an agent like a gene transfer therapy that is using a virus as the ability to bring its cargo, which in this case is the microdystrophin DNA, into the body. So we're hopeful that what we're doing is, in addition to the prednisolone or the prednisone that we're using to reduce the inflammatory response in the body, we're also now introducing sirolimus as another mechanism to reduce the body's inflammation risk.
Thank you, Crystal. Very helpful. Now we'll get back to the data presented. So I just wanted to get those two sort of overarching questions because I know they're on the minds and top of mind to many parents and patients who are considering this therapy. So on slide 23 and 22, you mentioned both the 10-meter walk run and the time to rise as increased. And in fact, on slide 23, you mentioned that the data between years 2 and 3 was double in terms of improvement, which gave rise to a 70% increase in terms of that excellent p-value and a slowing progression at 70%. Crystal, I'd like you, so I'm a parent coming to you and saying, what is 70% slowing progression? And what does that mean for me to interpret in my future, or my son's future, or for that matter, the patient coming to you saying the same thing? Can you translate this 70% slowing progression and 73% slowing progression in what that actually translates into the future of the natural history of this disease?
I think that's a great question. When we look at this data, what I can say is that it reflects where our boys have come from an average age of essentially 6 to 9. And so over that time period, we've seen a 70% slower progression rate. Now, what that means for the future is something that I'm very optimistic for, but we're going to have to take this year by year. And I think it will become incredibly meaningful over the course of the next few years, where we start to be able to tabulate data on time to loss of ambulation. Now, I would interpret as a clinician looking at this data that perhaps we will see a delay in time to loss of ambulation. And I'm hopeful that that's what we'll see. But we won't know that until we get this data. I think what this shows us when these boys are at an average age of 9, and is that we are seeing a clear distinction from what has been known to be natural history. And I think that this indicates, at least to me as a treating physician, that I really should be optimistic about the potential benefit for our boys who we have intervened with utilizing ELEVIDYS. And as I think James nicely pointed out, especially with some of the MRI data earlier, remember that we're seeing these physical changes, these time testing changes, well after the muscle itself has biochemical changes that we've been able to detect on MRI. That happens first before we see this physical slowing. And so there's some thought here that perhaps we should be intervening earlier and not delaying this treatment, understanding that the earliest age that our boys can get ELEVIDYS in particular is set at age 4 for right now.
Yeah, thank you. We'll get to the MRI in a few minutes, and I agree that there's a change that we can track through the fat fraction and infiltration of muscle, and it's also a predictive value. I guess in looking at this cohort of individuals, and this is part 1, so this is the initial active people who have received ELEVIDYS at year 1 at baseline, right? And then this is looking at that cohort. So do we know in years out, this patient group, have they continued on daily steroids, or have we differentiated the steroid regime that they're on because they were from baseline asked to increase their steroid for a period of time? Have they stayed on steroids? Have they added other medicines, like returned to if they were amenable to an antisense, or included any of the new medicines or more recently approved medicines such as Agamree or Duvizat? Do we have that data, and has that in any way helped with the data analysis, or is that a later analysis?
So I think that's a really important question, and it's one that we're still working our way through, if I'm honest. So we have looked at these data. I say that the majority of boys remain on daily steroids. We have seen a few transition to weekend steroids or to other regimens or other drugs like Plazicort. We have within Embark, you couldn't have another additional treatment like Givinostat, but within Expedition, so the final 3rd year of these data, you can add in patients, again, a handful of Givinostat added into a handful of patients. So that's the kind of qualitative answer, is that we have seen some changes, and I think that's inevitable as you follow individuals out multiple years, that other treatments are going to be, and I think that's a fantastic thing for the field, that there's the option to weave other treatments into the paradigm. I don't think there's an indication in terms of scale that it's likely that these changes have affected the results, but I can't say that definitively and answer the question really properly until we've done that analysis, and we'll come back to you.
Yeah, thank you, James, for that. I think probably since I entered this field, which was many years back, we've always talked as a community that would take a combination of medicines to halt progression. And so we're talking here with the ELEVIDYS data about slowing progression, which is really wonderful, but halting progression would mean, or likely mean, that we would add additional medicines. And that's why I asked the question. So I appreciate that you're going to look into that. And I'm guessing with the small number of patients here, that perhaps the few that would have gone on other medicines maybe wouldn't impact the data that you're showing us today. So thank you for that. On slide 18, we talked about natural history data. So can we go to slide 18, Wendy? Oh, I must have, yeah, there you go. Is that not slide 18? Thank you. So talk to us, and Crystal, talk to us about these natural history data that was utilized for this analysis. I'm very familiar with 4DMD and Synergy and the tadalafil study and Demand and et cetera. So when you characterize these patients, you've identified how you characterize them, but did you, in terms of weight and BMI and all of the outcome measures, but did you also genetically characterize them? So in general, in your exclusion criteria, it was 1 to 17, if I recall correctly, in this study, and after 59 excluded. So did you also include genetic characterization of the folks that were in this natural history data set?
No, so we didn't exclude on the basis of underlying genotype. Once you essentially build all of the other characteristics in terms of baseline function into the statistical model we use, the impact of the genotype is not significant. It doesn't affect the results, essentially. So we didn't have to exclude genotypes here.
All right, thank you for that. And in for DMD, so can you tell us how you do these natural, how you look at what age match controls are in terms of these are not the people in the active role, in the active part of for DMD. This is the placebo, or how do you pull these folks out so that we can understand how do you age match control those folks?
Sure. So we have a criteria, first of all, that any of the patients that we compare against have to have been on chronic corticosteroids for at least 12 weeks. We then look at the top end of the key kind of characteristics that we know that predict disease progression in Duchenne. So things like baseline NSAA and baseline time to rise. We look at those values from the ELEVIDYS patients, so we know what the minimum and maximum for each of those values is for the ELEVIDYS patients, and then we pick any patients that fall within that window out of these clinical trials, these natural history patients. And that gives us a patient population that looks pretty similar at baseline. And then you make it very similar by doing this propensity weighting thing, which is quite a complicated statistical thing, but ends up really basically just giving more value to the external control patients that most look like the ELEVIDYS patients. And you do that repeatedly, and you end up with data that looks very, very similar at baseline, which you can see actually on slide 20 if you go on one, Wendy. So by selecting this cohort of patients from the external controls and then propensity weighting it, you get this data that very closely match on what are the key prognostic markers for how we think that Duchenne is going to behave subsequently. So that's the NSAA there. You see that the ELEVIDYS patients were 23.3 at baseline. Our external control after this process was 23.5. 3.51 for time rise, 3.52 for the external control. And I won't bore you by reading the table out, which you guys can all read yourself. But it's essentially a two-step process that then I think leads to what is a very closely matched comparator set.
James, I really appreciate the explanation, because I think in other studies, to include this one, we're using age match controls. And I think for our community to understand how those studies are done, and certainly for regulatory, for FDA and EMA to look at these age match controls and to be able to utilize them. I'm hopeful that one day, I'm not sure how soon, but we could get away with placebos looking at age match controls so that we could define benefit and risk of any study going forward, and maybe get earlier results and more earlier approvals. So thank you very much for that. And coming back to slide 14, this is the 301 study. I think, James, just for this audience, there have been so many different studies in EMBARK. Can you just refresh us again about the 301 study as compared to the 302 study and other studies, so it's very clear in our mind what this study was about and what part we're talking about, the part one today?
Absolutely. So the 301 study, or EMBARK, was a phase 3 study. So that's a study that is designed to confirm efficacy and safety, and is what is usually required in the United States for a traditional approval or full approval elsewhere in the world. This looked at individuals aged 4 to 7 at baseline, excluding mutations in the 1 to 17 region, as you mentioned previously, Pat, and excluding patients who were antibody positive to RH74. It was around 125 patients randomized either at random, were either given the treatment, ELEVIDYS, or given a placebo in a blinded fashion. So nobody, including ourselves, knew who got what, and then had a number of functional assessments over the course of a year. The primary number one sort of assessment in the hierarchy was the North Star assessment, ambulatory assessment, and then we also looked at timed function tests. We defined effectively key secondary outcomes of time to rise and 10-meter walk, run. And so this study versus placebo was unblinded 1 year, where whilst the primary endpoint of North Star ambulatory assessment wasn't statistically significant, although numerically superior for ELEVIDYS, we had consistent benefit across all of the timed function tests, which I think is a statement that supports what you said earlier, Pat, around about 1 year, I think probably being too short in this disease to look at an outcome. The end of 1 year, our patients who were treated with ELEVIDYS received placebo, and the patients who got placebo got ELEVIDYS, and then the study continued for another year, and that's the full 2 years. What we're looking at today is the patients who were treated with ELEVIDYS at the very start. So that's the 63 in the purple box at the top left of this diagram, and that's following those patients out for 3 years following that initial treatment.
Yeah, thanks, James. And you use the SV95C in this study as well. Do you have any data on the SV95C and how that mirrored or matched up with the 70% slowing and 73% slowing that you've talked about?
So SV95C was statistically significant, showed benefit for ELEVIDYS at 1 year. We didn't continue that assessment into the expedition, so we don't have any more data on that.
Right, and then the same for the forced air climb. You were really focused on the.
Yeah, sorry, Pat, I didn't talk over you. So four-step climb is a slightly different story. So we do assess four-step climb in expedition. I don't have the data to present today. What we don't have with four-step climb is an external control to match it to. So that data is less frequently collected in natural history studies. So we can't find the data to provide the comparator that we did with time, 10-meter walk, run, and time to rise. I think at some future point we can certainly show the data without comparison, but I don't have it today, I'm afraid.
Pat, that reinforces the point that the external control is not just age match. There's several parameters that factor into matching the external control to the study population.
Right, Wendy, because obviously in most of those studies, Synergy, 4DMD, et cetera, they didn't have the SV95C, although some of them did use the forced air climb. I'm not sure which ones, but probably more difficult to do an age match control on that. If we go to slide 16, let's talk about cardiac MRI. I think it was slide 16, if I remember correctly. Yeah, so James and Crystal, at baseline, are these individuals in the study, did they have, what was the cardiac baseline you were looking at? Did they have to have an EF of a certain, of not less than a certain number? And did they have other tests that would lead you to believe that their cardiac MRI was going to be pretty stable throughout the course of this study?
They were excluded if they had a cardiac function that was lower than, I believe, James, correct me if I'm wrong here. I believe it was that it needed to be above 40%.
40%, yes, Crystal, sorry.
And so as you know, Pat, and those on the line, ordinarily our patients at this particular age, it would be less likely that they would have ejection fractions below that. But there are certain circumstances where we do see that with earlier incidents of cardiac dysfunction. And so the goal was obviously to compare similar patients to similar patients. And so they were excluded if they had significant cardiac dysfunction. So you can see that at baseline, these patients had good cardiac function, and that remained for this period of time post-treatment. as well. And this will be something that will be important for us to follow over time, is looking at their ejection fractions, looking at, for those that can undergo cardiac MRI, looking at their ejection fraction in addition to if we can get things like the degree of late gadolinium enhancement. I think these are going to be incredibly informative, especially as our boys age, but they're still in a very young age group right now. So time will tell.
Yeah, thank you, Crystal, for that. And just to explain to the group on the phone, just could you give us a little insight into LGE, because I think that might be a new term or an unfamiliar term to the group that's listening here.
Absolutely. So LGE, I think, is something that neurologists are even newly getting comfortable with as we assess our guys with Duchenne over time, both clinically and as part of research studies as well. So the way that I like to describe it to my families and my boys that are in clinic is that we're taking a picture of the heart with an MRI, which is incredibly detailed. And we're not just looking for the percentage of squeeze, and so the strength of that heart muscle, but we're also looking for the degree of scar tissue. And what we can see is that scar tissue is not necessarily going to show up on ultrasounds like with our echocardiograms. It really requires us to be able to give something called IV contrast through an intravenous line, so through a peripheral IV that's placed usually in the arm, and they will give this contrast. And they'll see what areas of the heart light up. And those areas that light up indicate that there is some scar tissue formation. And so we can track this over time. Our colleagues in cardiology are getting better and better at how to quantify this. It's not been perfected yet, but we're working hand in hand with them so that we can better understand how does this scar tissue change in untreated guys with Duchenne, and how might it change or be impacted, hopefully for the better, if they were to receive therapy.
Yeah, thank you for that, Crystal, because I think we are hearing about LGE and wondering exactly what that does. And I appreciate your explanation because I think it was very clear and very helpful. So at baseline, did any of these children have LGE that you know about, or did they have to have a pretty clean MRI with a stable LVEF, as you talked about?
So
Unfortunately, we didn't actually use late gadolinium enhancement in this study. That was based on levels of LGE in children of this age are very low. Now, that doesn't mean that it wouldn't be good in retrospect to have a baseline, but very unlikely that many, if any, of these studies children be LGE positive at the ages of 4 to 7 when they were first enrolled into this study. And contrast, although I think my cardiology colleagues would tell me is very well tolerated, it does have some side effect risks. So on balance, we didn't put LGE into this study. LGE assessments are being done in the follow-on expedition study and are being done in our studies in older patients who are much more likely to have that finding at baseline.
Yeah, I don't disagree. I don't disagree that I don't disagree at all that you're unlikely to see an LGE here, James. But I do think, we as a community are seeing are hearing about children with really concerns about their cardiac MRI and LGE. So I wanted to raise it just so that to level set with the community that this was a younger population, likely a pretty good cardiac MRI in this group. What we do see on cardiac MRI is sometimes some strain at this age, but that's typically, at least the action or data suggests that that's about what we see in this age group. But as they get older into this 9 to 12 age range, then you might see changes. And I think that is, to Crystal's point, the reason to continue to follow these children, because we are very hopeful that there is a protective factor of gene therapies on these children's heart, because the heart is a muscle too, and we want to make sure their improvement in terms of function and preservation of function, that their heart's good enough to last. I guess that was perhaps a very tangible way of suggesting that, but that's why I think as a community, we're very concerned about that. I don't know, Matt, I don't know if I have an echo, but hopefully not too terrible of an echo. I've tried to decrease the sound here. So thank you for notifying me about the echo. On slide 11, you talked about the natural history of this disease. I think what what we've heard in this community is that these children are, you mentioned the downward pressure, certainly of the disease and its progression. And what we're trying to do is stabilize that and improve it. Peak age at 6 years old, is that what the data, the natural history data is saying? Just for the community, and what we hear sometimes is sort of the period at which time there's a stabilization and then decline is usually between 7 and 8 years old. But I wonder if your data is suggesting that that's different and it's about 6 years old where we see that plateau and then start to decline. Can you just give us a little bit of color on that one?
Yes, I mean, I can certainly provide my reference to this, which comes from the Synergy study, which has a mean age peak performance at I think 6 and a little bit, 6.2 or something like that. Obviously, it's a very heterogeneous, very variable disease, and I think talking about a window for that makes a lot of sense. But if you pick one statistical value, it's around about 6 from I think the most extensive natural history studies. But Crystal, please feel free to correct me.
No, I think that that's the data I'm familiar with as well. I think it peaks at 6.3 and then we see a slow decline thereafter, at least the data that I'm familiar with.
Sorry, Crystal, does that depend on the steroid regimen that you see? Is there variation? And I'm just curious about that. I'm not going to disagree. I've seen the 6 and I've seen differences, but I wonder how the exact steroid regimen impacts this or not.
So I think that that data comes from folks that were on corticosteroids on a daily basis, if I'm remembering correctly.
Thank you so much for that. Crystal, now I'm going to put you online here. And so as a patient coming into you, and we've talked about, you mentioned earlier, treating is going to be optimal, we think, just in terms of getting the greatest degree of preservation of muscle function here. But I think, could you as a clinician talk to us about the need to treat, not just recognizing that earlier is better, and I think in any disease, earlier is better. But what are you seeing over time? What are you seeing over time with all ages? You've mentioned at the beginning, James mentioned that Sarepta has treated patients from 2 to 24 years old. Can you just give us your insight as a physician? What are you seeing? What are you thinking? And when we toss around 70% slowing, that seems like I would raise my hand and say, I'll take that forever. But how would you counsel me as a mom of a 9-year-old coming to you at this moment?
Thanks for that question, Pat. I've had the pleasure and the honor of taking care of boys who were both 2 and giving them this therapy, and the gentleman that was 24 at the time of therapy. And I think that the dialogue that we had at the time has continued to be very similar, if not more optimistic now than when we had those dialogues at the time of treatment. And I think the thing that I focus on with families when I am talking about options, because we're so fortunate to have a variety of options these days, including clinical trial, is that I think we need to first and foremost try to establish a foundation of dystrophin restoration if there is the capacity to do so. And what I mean by that is the ability to try to optimize that dystrophin protein within the muscle. And so there are going to be some boys that will not be able to be accomplished, whether it's because of their genetic mutation or the fact that they have antibodies present. But if they do qualify to receive a dystrophin restoring therapy like gene transfer therapy, that for me is going to be a foundation. And I say a foundation because I think we need to build on that. I think this graphic that you have up on the screen right now really is a great one to show that our goal is stabilization for as long as we can. And I want to see that natural history distinguish itself from our treated cohort. And I want that delta, that difference between the 2 groups, to get larger and larger and larger over time. I think that's going to start with dystrophin restoration, upon which we can build with some of these other opportunities we have in the therapeutic space now. And so that's kind of how my dialogue with families goes now. I don't know that has changed. And I think with this data here today, I'm even more encouraged by the potential benefit as we move forward. and as we move perhaps earlier in our treatment journey.
So following up on that, Crystal, and I thank you for that, because I think that, gene therapy is here and we're very excited. Some of us have waited a very long time to see it, having thought, how hard can this be 30 years ago? Turns out it's pretty hard. It took 30 years. So there's that. And we're really thrilled to be in the era of gene therapy to restore dystrophin expression, a microdystrophin in this case. So my question around that is really thinking through, you mentioned, and James has mentioned, the 70% change and how the time, the 10-meter walk run, as well as time to stand, has really increased in the delta. So at a certain time, and we don't have the data, but do you suspect that will plateau as well? Or do you suspect that it will continue to increase the delta over time? So would you expect 9 might be a plateau of 3 years out, or I know you don't have a crystal ball. I wish we all did, but how do you see that going in terms of going further out as a mom? What could you tell me about that? What do you suspect? What do you know? What do you not know at this point, and what do we have to learn?
I'll be honest, Pat, as I was thinking about the potential for this 3-year data to come out, I had an expectation. My expectation was that I was going to continue to see that this difference between the untreated external group would be more notable than even at the 2-year data, because we unfortunately know that our boys, as they get older, they have these declines. And I've witnessed in clinic and in clinical trials that the boys that have received treatment, they're changing in a different way and at a different rate. And so I was very pleased to see that my expectation was met as I look over this data that James has shared with us today. And so my hope is that we continue to see that continue. I don't know that I can expect a plateau of that difference or when that may be. All I know is that as we continue to keep our boys under the 10-second mark for the 10-meter walk, perhaps under the 5-second mark for their time to rise. These are things that are all going to impact our anticipation of time to loss of ambulation. And we know that once we see that, we can also anticipate changes in respiratory status and perhaps changes in cardiac status. So these early indicators I find very meaningful for long-term clinical outcomes. And so I'm optimistic that we'll see that as the years go on.
Yeah, thank you very much, Crystal. And this data comes from the study itself, not from the commercial use. So I'm sure in your phase 4 data, we will learn, excuse me, even more, as you've talked about how many have been treated with I think it was 240 in the context of a clinical trial, and then many more in the context of commercial. So I think this is very good data. It's very encouraging to see that gene therapy, as we look forward to gene therapies coming online and looking into the out years, that this continues to preserve muscle function and hopefully cardiac function, as well, as we'll learn in the out years as well. So I can't thank you enough. And with that, Wendy, we're getting about time. I will turn it back over to you to close out. But thank you to Sarepta. Thank you to James. Thank you to Crystal for taking time out of your clinic to really come to the community and explain what you're seeing, what we know, what we're going to learn in the future, and how we'll move forward together. So Wendy, back over to you.
Thank you so much. And Dr. Proud, thank you. You have a very, very busy clinic, and we certainly appreciate the time that you've been on this call today to share your experience. And most importantly, we want to thank everybody in the community. Research like this doesn't happen with families and loved ones and advocates supporting everybody along the way. So as Pat said, gene therapy in particular is really hard, and we couldn't do this without everyone working together. So huge thanks. You can send any questions to advocacy@sarepta.com, and we look forward to getting together again soon.
Thank you all for joining us today. And with that, we'll conclude the webinar. It will be posted within a few days. Once we get everything together and the approvals in line, you'll see this back online and can review the webinar in detail. As Wendy said, if you have questions, please send them to advocacy@Sarepta or send them to PPMD, and we're happy to get back to Sarepta and make sure all of your questions are answered. And with that, wishing you a good rest of your day. Thank you so much.
Thank you.